Good afternoon, all of you. In today's lecture, we'll start the unit number 5. That first point in unit number 5, that is the microbial spoilage or the spoilage and the types of spoilage. Generally, the question asked in examination on spoilage, that is write in detail about types of spoilage, then or give the types of microbial spoilage for the pharmaceutical product. Yes, I am repeating the question on this. Write in details about the types of spoilage, or they explain various types of spoilage, or the give types of microbial spoilage for pharmaceutical products. Generally, the microorganisms are present in different parts of environment. For example, soil, then sewage, then water, then animal waste, food and air, and which are responsible for contamination of pharmaceutical product, which is called as spoilage. I am repeating here, the microorganism generally are present in different parts of environment. For example, soil, sewage, water, animal waste, food and air, and which are responsible for the contamination of pharmaceutical products, which is called as spoilage. The spoiled pharmaceutical products can cause the spoiled pharmaceutical product can cause first, that is infection. For example, typhoid fever by contaminated thyroid tablet, then lethal reaction, then deterioration of product or the loss of activity of product. And this deterioration of product is detected by, basically there are two changes. First, that is physical change, and second, that is chemical changes. Here the spoiled pharmaceutical products can cause, first, that is infection. Example, typhoid fever by contaminated thyroid tablet, then lethal reaction, then deterioration of product or the loss of activity. And this deterioration of product or the loss of activity, it can be determined or detected by, first, that is physical changes, and second, that is chemical changes. We will see the types of spoilage. First, contaminated pharmaceutical product can cause infection. In that, the main source of microbial contamination in pharmaceutical products are, first, raw material. For example, the contaminated water used during manufacturing. For example, the contaminated water used during the during the manufacturing can cause spoiled pharmaceutical product that causes the cholera. For then here we can see the various dosage form and reason of spoiled and reason of scots. First, tablet despoiled by salmonella species, and cause of this, that is salmonella infection. Then eye drop, that is pseudomonas aeruginosa, causes or cause eye infection. Then for antiseptic solution, pseudomonas species, and cause, that is septicemia. Then for ointment and creams, is gram negative bacteria, and for that cause burn or dermatodes. Then for IV medicines, for that there is candida species, and for that cause of fetal septicemia. Then second step, that is steps in preparation of pharmaceutical product. Generally the pharmaceutical products can be spoiled by various types of bacteria. Generally the pharmaceutical products can be spoiled by various types of bacteria. For example, mesophiles, thermophiles, psychrophiles, and psychotrophs. Then that is then physical and chemical deterioration of the product, that is the loss of activity of product. In that, the matter are more sensitive for deterioration, in which they undergo the chemical reaction. For example, use of animal and vegetable drugs. Because of this material act as nutrient media, and which increases the risk of destruction by the microbes. And the rate of deterioration, it means the loss of activity, and this rate of deterioration is depend upon, first, that is molecular structure, then environmental properties, physical properties and chemical properties, then types of microbes, then quantity of microbes, and presence of metabolite. Yes, this rate of deterioration is depend upon first, that is molecular structure, then second environmental properties, then physical properties, and third that is chemical properties. Then it is also depend upon the type of microbes, then quantity of microbes, and the presence of metabolite. These the rate of deterioration, that is, it means the loss of activities depend upon these all factors. Then third, that is effects of microbial attack. That first, that is microbes, it attack on the formulation of ingredient and as well as the formulation procedure of the substrate that is essential for the energy production as well as the energy replication. Yes, first that is microbes, it attack on formulation ingredient as well as the procedure substrate and the produce substrate that is essential for the energy production as well as the energy replication. Then it contaminate the product by two ways. First, that is on moisture surface film, and second, that is unevenly distributed in bulk of viscous. Example of first, that is on moisture surface film. Example, that is film, and second, unevenly distributed in bulk of viscous formulation. For that example, that is cream. Then spoilage indication, that is first, that is unpleasant smelling, then unpleasant tasting, then discoloration due to the pigment by microbes, then change in viscosity due to the depolymerization of suspending agent or the depolymerization of thickening agent like tragacanth, acacia, and CMC, that is carboxymethyl cellulose. I'm repeating the spoilage indications. First, that is unpleasant smelling, then second, unpleasant tasting, then discoloration due to the pigments by the microbes, then change in viscosity due to the depolymerization, then depolymerization of suspending agent, then depolymerization of thickening agent. For example, I would like tragacanth, acacia, and CMC, that is carboxymethyl cellulose. Then the production of slimy viscous mass due to the microbial polymerization of sugar or the surfactant in formulation. For example, spoilage in syrup, then spoilage in shampoo, and spoilage in cream. Then greatest texture in creams due to the growth of fungi, then change in pH due to the production of metabolites. Example, that is acidic, basic, or the gases. Then next, that is pharmaceutical ingredients susceptible to the microbial growth. In that first, that is therapeutic agents. The pharmaceutical ingredients susceptible to the microbial attack. First, that is therapeutic agent. In microbial spoilage, these active therapeutic agents are metabolized by microbes and converted into the less potent drug which cause the loss of activity. For example, alkaloids. In example of alkaloid, that is morphine or the atropine. Then second example, that is analgesic. The examples of analgesic: aspirin, paracetamol, thalidomide, and barbiturates. I am repeating the examples of alkaloid, that is morphine, and second example, that is atropine. Then example of analgesic, that is paracetamol, aspirin, thalidomide, and second last, that is barbiturate. Then example of an example, so that is eye drop, in which the active ingredient is atropine metabolized by fungi. Then for penicillin injection, in which the penicillin inactivation by beta lactamase bacteria. In eye drop, or in which the active ingredient is atropine and which is metabolized by fungi. Then penicillin injection, in which the penicillin inactivation by beta lactamase bacteria. Then for steroid, it is metabolized by fungi on damn tablet. Then aspirin hydrolysis by esterase producing bacteria. Then chloramphenicol, it degrade by chloramphenicol acetylase. It degrade by chloramphenicol acetylase producing bacteria. The next, that is surface active agent, or it is also called as what? That is surfactant. In that, first anionic surfactant, they are stable in alkaline pH. Then examples, alkali metals, amine soap of fatty acid. And when this anionic surfactant diluted with sewage water, it easily degraded. Yes, example of anionic surfactant, alkali metals, amine soap of fatty acid. And when this anionic surfactant is diluted with what? That is sewage water, it get easily degraded. Then, for example, alkyl benzene sulfonate metabolized by two step. First step, that is oxidation of terminal methyl group, and second, that is beta oxidation of alkyl chain fusion of aromatic ring. Yes, the metabolization or the alkyl benzene sulfonate is metabolized by two ways or the two steps. First, that is alpha oxidation of terminal methyl group, and second, that is beta oxidation of alkyl chain or the fusion of aromatic ring. Then second, that is non-ionic surfactant. It is metabolized by various microbes. These non-ionic surfactants are metabolized by various microbes. For example, alkyl polyoxyethylene alcohol. And the rate of metabolism is depend upon, first, that is chain length, then second, branching in chemical structure, then ampholytic surfactant and cationic surfactant. I am repeating here, the rate of metabolism is depend upon what? First factor, that is chain length, then second, branching in chemical structure, third, ampholytic surfactant, and last, that is cationic surfactant. In that first, chain length, if the chain length increases, attack decreases by microbes. Then branching in chemical structure, if the increases branching decreases the attack by the microbes. Then ampholytic surfactant, example phosphatides, then alkyl amino substituted amines, they are biodegradable. Then cationic surfactant, they are slowly degraded by microbes, so it's used as a antiseptic as well as preservative. The next, that is polymer. For the degradation of polymer, for example thickening agent or the suspending agent, the microbes produces the extracellular enzymes which carry out the depolymerization. Then example of enzymes and the substrate that degrade, that is example of enzyme, that is amylase, and substrate that degrade, that is starch. Then for pectinase, that is for that it is pectin. Then cellulases, then for cellulose and CMC, then dextranase, that is dextran, and protease, that is protein. Then the degradation governed by molecular weight. Then lower molecular weight, for example polyethylene glycol, increases the degradation by the oxidation. Then polymer highly resistance for degradation, example nylon, then polystyrene and polystyrene and polyester. Then next, that is humectant. What is humectant? It means which prevent the water loss or it maintains the moisture but easily get degraded due to the low molecular weight and the low concentration. But degradation reduces at high concentration. For example, glycerol and sorbitol. Then for fats and oils, they are commonly used for formulation of oil water emulsion. These fats and oils are commonly used for oil water emulsion, but the commonly spoiled by growth of fungi in oil water phase in emulsion. For example, triglycerides after the lipolytic breakdown to form a glycerol and the fatty acid. It means triglycerides, it undergoes the lipolytic breakdown It gives or to form like glycerol and the fatty acid. After the beta oxidation of what? The beta oxidation of glycerol and fatty acid. After the beta oxidation of glycerol and fatty acid, it gives or it produces odoriferous ketone. Then sweetening agent, flavoring agent and coloring agent. In that first sweetening agent, example sugar, highly support growth of microbes, the microbial spoilage at less concentration. But high concentration they reduce the microbial spoilage. But in high concentration they reduce the microbial spoilage. Then second, that is flavoring and coloring agent. Stored in aqueous stock solution are rarely attacked by microbes. So far, the reduction of spoilage freshly or use of alcohol for the preparation of or the prepare flavoring agent or the coloring agent which are used in or which is used for manufacturing. For example, amarnath, then tartrazine and peppermint and the peppermint water. The microbial spoilage commonly due to the Pseudomonas aeruginosa. This microbial spoilage is commonly due to the what? That is Pseudomonas aeruginosa. Then last, that is preservative and disinfectant. At low concentration of organic preservative or this disinfectant, then it get metabolized by fungi and bacteria, that is generally the gram negative bacteria. Then example, quaternary ammonium antiseptics, they get degrade of the spoiled by Pseudomonas species and it causes the various infection, that is eye infection. I am repeating, the preservative and disinfectant at low concentration of organic preservative or the disinfectant, it get metabolized or the metabolized by fungi and the bacteria, that is gram negative. The quaternary ammonium antiseptics, that is chlorhexidine 4 hydrobenzoate ester, which get degrade of the spoiled by Pseudomonas species and causes the various infection, for example eye infection. So this is all about the spoilage and the type of spoilage. I will stop here. Next part we will see in next lecture.